Hi, I am Mrs. Sloan, and this is a video intended for my AP Biology class, but everybody's welcome to watch it. And so we're going to continue on talking about the cell cycle, and we're going to talk about it when good cells go bad. Remember, we're under the big umbrella right now about talking about cell communication, and cancer is when you get the wrong signals, so you get the wrong metabolic activity. All right. And for those of you that are new to my presentations, the notes are in the descriptors down below, and column 1 is all about filling those notes in, like my students do. And then column 2 is left blank, and that is there so that you could add pictures or things in from the diagram. So we have already gone over in video 1 for chapter 9. We have gone over the cell cycle, and we have learned mitosis. So now what we're going to do is look at when you have a abnormal cell cycle and how that relates to cancer. All right, so good cells versus bad cells. You can see them right underneath the microscope, no problem. Sadly to say, my mother had chronic lymphocytic leukemia, and she passed away from that. And when we were at Stanford Hospital and they were treating her, they removed some of her bone marrow from her hip. And I was right there with them when they did these treatments. And I looked in the microscope with the doctors right there in the room when they looked to see if the treatment is working, and you could see the cells were completely abnormal from what you would expect to see. You can see it very readily externally from the cell, the way the nucleus looks. So we're going to talk about those specific structures that look different, but also what behaviors change with the cell as well. So when you have a cancerous cell, you have loss of normal control. And it's said that probably every single day you have a cancer cell, and every single day your body takes care of that abnormal cell and destroys it, right? And remember, cellular suicide is apoptosis. If we don't catch that abnormal cell, remember the checkpoints we talked about when we learned about mitosis. If it's not caught, fixed, repaired, or if you can't repair it, if it's not eliminated, then that's when you get that uncontrolled growth. Now, normal cell characteristics, okay? If you take a look at this diagram right here, so you can see the cells are kind of filling in. They have a couple of characteristics. One is anchorage dependence. Most cells have to be attached to something. Obviously, exceptions to that would be like red blood cells and white blood cells, right? Because they're moving all throughout your blood. But normally cells have something called anchorage dependence. So that's one characteristic of normal cells. And then they have something called density dependent inhibition, which means when they get crowded, when they can fill a neighboring cell, then they're like, oh, we're crowded, we should stop dividing. So those are characteristics of normal cells. Here you can see they removed a few cells, and then mitosis occurred again and it filled in. So on your notes, we are on 9.4, the cell cycle and cancer, which is 4.7, right, in the AP unit guides. Density-dependent inhibition is the phenomenon in which crowded cells stop dividing. So another name for that is contact inhibition, if you want to add that to your notes, contact inhibition. And another item, a characteristic of normal cells is anchorage dependency. Normal cells must be attached to a substratum, like the extracellular matrix of tissues, to divide. So they have to be attached before they divide. If they become free floaters, they don't. So in a neoplasm, which is an abnormal growth of cells, and you have that on your notes right there, you could develop a tumor. Now, why that occurs is because cancer cells do not exhibit anchorage dependence. If they get separated, they'll keep dividing anyway, and they lose their contact inhibition, their density dependent inhibition. So on your notes, you already have the definition of a tumor. Let me go right here. Okay, so if it's a benign tumor, it's encapsulated. Oftentimes you will see, to check to see if you have a tumor. So breast exams would be something you do, where if you notice an abnormalities, or in testes, if you notice a growth coming up, that you want to go in there and get that immediately, because it can gain the ability through mutations to metastasize and spread throughout your body, then it is malignant. So you want to go in and get those tumors when they're still encased, when they are a benign tumor, because once they spread, it's very difficult to control it from there. So benign benign tumor is not cancerous. It's encapsulated. It has not invaded adjacent tissue. It has not invaded adjacent tissues. A malignant tumor has invaded, or it has the ability to spread. It has the ability to spread. Because if you look right here, look at this tumor right here. Now, first of all, one of the things it has to do is it has to be able to gain the ability to go from being a benign tumor. It has to gain the ability to digest what's called the basal lamina to get out of that. And here you can see it's continuing to spread. Once it gets in your lymphatic vessels or it gets into a capillary, this is metastasis. So it's spreading everywhere. And then it's like, it can get off. Think of your blood supply as like a train system. It can get off at any station it wants to and set up camp in all of those different areas. So metastasis is cells separate from a malignant tumor and enter the blood or lymph vessels and travel to other parts of the body So why is it doing that? Okay. Why are these good cells going bad? Well, probably because you have some mutated genes. Remember, your DNA is your code book. And if something happens to damage your DNA, I don't know, maybe smoking cigarettes, okay, or vaping. If something happens in your environment that could alter that DNA, you could get wrong genes, which then code for wrong proteins. And those wrong proteins could be part of how your cell cycle is controlled. So if they mutate, that's going to be your problem. So on your notes, cancer, cellular growth disorder that results from mutations of genes, G-E-N-E-S genes, that regulate the cell cycle, and you have loss of control. So abnormal cells, so they defy the normal regulatory procedures you have in your cell cycle, and then they have the ability to invade other areas. All right. Carcinogenesis. Genesis means beginning. So carcinogenesis is the beginning of cancer. Now, most cancers are fairly gradual. They could take years and years and years and years in order to develop because they have to accrue multiple mutations until they become cancerous. So carcinogenesis is the development of cancer, which is usually gradual and can take decades. Retinoblastoma, cancer of the eye, is pretty quick. And then I'll talk to you about that a little bit more in a minute, okay? So when you want to compare and contrast characteristics of cancer cells versus normal cells, here's just a little list right here, and I'll help you on your notes. So cancer cells will be differentiated. Normal cells will be differentiated. Cancer cells are non-differentiated. They don't have their specificity. They have really abnormal nuclei. They don't do apoptosis. Remember cellular suicide? They don't do it even though they're broken. They don't exhibit contact inhibition. They're very disorganized, multi-layers, and they can spread, okay? And I'll show you some more on that. So on your notes, they lack differentiation. They are not specialized. They seem immortal. All right, so here you can see, let me get myself out of the way here. You can see, whenever you see blue slate, that would be if I was in my classroom and I have people who sit in blue chairs and slate gray chairs and they ping pong back and forth, sorry. So this is our new normal. If you compare and contrast, here is a normal cell. Here's your nucleus, right? Here's your cytoplasm outside. That would be normal. Okay, and your nucleolus. But look at the cancer ones. They could be multinucleated, have very little cytoplasm, multiple and large nucleoli because they're cranking out so many ribosomes because it's out of control. Very coarse chromatin. Remember, chromatin is the association of DNA with proteins. So on number 2, abnormal nuclei, and then you have the notes for that, right? They're enlarged, or abnormal number or deleted or duplicated portions of the chromosome. And that's called gene amplification when you have multiple copies like that. All right. They do not undergo apoptosis. Remember, let me see if I can do it again. Apoptosis is cellular suicide. Most cells go about 50 divisions and then they're done. And if they have abnormal DNA, then they get killed off. But these do not, okay? Normally cells enter the cell cycle, or cells leave the cell cycle, right, and go into G0 after about 50 times of replication. Now, angiogenesis, this is super, super creepy. Now, when you have a benign tumor, okay, let's say it's encapsulated and closed, oftentimes the reason why it can't metastasize is because you have more cells growing there than can be supported by the nutrient supply. So that sometimes keeps it from metastasizing. But what some cancer cells have done is they have gained a mutation which causes blood vessels to grow towards them. It's like, come here, little blood vessels. And so the blood vessels start growing towards, that's called angiogenesis, the beginning of those blood vessels. Those blood vessels go and grow towards the tumor, and all of a sudden now those cancerous cells have a source of oxygen and nutrients and a way to get rid of their CO2 and waste. And then it's like the light is green. Then they start spreading, and they have a way to spread to other places because they have this great transportation system of your blood vessels. So on your notes, they form tumors. They do not display density-dependent inhibition. That's number 4 underneath characteristics of cancer cells. Number 5, they undergo metastasis and angiogenesis. I gave you the definition for metastasis. New tumors form which are distant from the primary tumor. And then B is angiogenesis, which is this slide right here, when tumor cells acquire additional mutations that allow them to direct growth of new blood vessels into the tumor. All right, so the origin of cancer. So it's going to be due to mutations, but this is the big ticket you need to get, right? Genes control your proteins. There are 2 big categories of genes, and you know what? Both of these categories have to fail in their job in order for you to get cancer, okay? Both have to fail. The first category of genes are called proto-oncogenes. Now, they're named after something they could become, which is an oncogene. And you know, if somebody has cancer, they go to an oncologist, right? That's where that's coming from. Proto-oncogenes are in a position of power. And like you might have a general that's in position of power because it's commanding all the troops, right? And if that, in a position of power, if that general goes bad, it controls so many other things that it can cause massive disruptions, okay? So those are proto-oncogenes. The other category of genes that we'll talk about are tumor suppressor genes. What tumor suppressor genes should do is they should take out the bad general, okay? They should take out the bad cells. So in order for you to get cancer, both of these gene categories have to fail. Proto-oncogenes mutate into oncogenes. They become bad generals, and those bad generals are not taken out by tumor suppressor genes, whose job it is to stop the bad generals, fix the bad generals, and if they can't, then kill them because they are in a position of power. So let's take our analogy here, and let's start with proto-oncogenes, all right? So proto-oncogenes, if it mutates, it will become an oncogene, and an oncogene is bad because it causes cancer because it will facilitate the cell cycle. So let me give you an analogy of this. This would be like if you're, driving in your car, right? And you're coming to an intersection, and normally you're like, okay, there's enough cells here, contact inhibition, let's stop, right? But this is like if you were unable to take your foot off the gas. It's like your terrible friend next to you reaches their leg across and jams your foot on the gas pedal, and you cannot take your foot off the gas, and so you would crash into that intersection, right? That's what an oncogene does, because it says, go cell cycle, go cell cycle, go. It's facilitating that cell cycle. Now remember what I told you about here, right? An assassin gene, right? Tumor suppressor genes. How could you avoid crashing? Yeah, maybe you can't take your foot off the gas, but maybe your other foot could do what? Step on the brake, right? So in order for you to get cancer, you have to have a friend pushing your foot onto the gas, and your brakes have to fail. The tumor suppressor genes have to fail as well, and then you will in fact crash. Most of the time when you have a cell like this that has gone bad, right, your tumor suppressor genes, your brakes work, and they prevent you from crashing into that intersection. So proto-oncogenes, these code for proteins that promote the cell cycle. promote the cell cycle, and prevent apoptosis, because they're like saying, go, 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 and prevent apoptosis like a gas pedal on a car. So they're a stimulatory pathway. When they mutate, they become oncogenes and cancer-causing genes. Now, how many of those, let's see, here's an example. Okay, here's your normal cell. It's a proto-oncogene. I don't know if you can read that. It's tiny. But let's say UV radiation causes this gene to mutate. Now it's an oncogene. Now it is a cancerous phenotype. So now normally you would need like a growth factor to stimulate, like a growth factor received at a receptor in order to stimulate the cell cycle. But due to this cancerous gene forming due to, let's say, radiation from the sun, now all of a sudden you don't need the growth factor. This cell goes through the cell cycle irregardless. Now, 100. You have 100 different categories of proto-oncogenes. Okay, that's a lot of genes that could go bad. So on your notes right there, there are over 100 potential oncogenes. Over 100 potential oncogenes that could go bad. Now, one of those is called the RAS gene family. And I always remember this one. RAS is like stepping on the gas, right? Like, okay, RAS is like stepping on the gas. So normally you need a growth factor to bind to a receptor, which then triggers the RAS gene, which then, right, signal transduction pathways like we've learned in this unit, facilitates transcription of this gene expression that facilitates the cell cycle, okay, for something, let's say a cyclin or something that's going to facilitate that cell cycle. But if you have a mutated RAS, notice it is still sending that signal, bad signal, sending it and expressing it, even though there is no growth factor here. So on your notes, for RAS gene family, this is associated with lung cancer, colon cancer, pancreatic cancers, leukemia, lymphoma, right? And thyroid cancers. That's a lot of cancers just from a bad RAS, because it's saying go cell cycle, go. And that's why you get the breast cancer, okay? Now, specifically, not with breast cancer. I'm jumping ahead. Here we go. With the breast cancer gene, you've heard of BRCA1, BRCA2, right? These are mutated versions of this, right? And these can be carried by both men and women, right? Predispose you to breast cancer. And you have that in your notes as well as ovarian cancer, okay? So proto-oncogenes, these are facilitating the cell cycle. And hopefully you can step on the brakes using your tumor suppressor genes. But if tumor suppressor genes mutate, okay, then your brakes are failing. And if you lose your brakes, you're definitely crashing, and that would be cancer. So tumor suppressor genes code for proteins that inhibit the cell cycle and promote apoptosis, like brakes on your car. So they prevent the cell cycle when DNA is damaged, or another thing they can do, they do both actually, is they prevent the cell cycle when DNA is damaged, and they promote apoptosis. They promote that cellular suicide. All right. And for this, there are about 6 identified. Okay. 6 breaks. All right. So here, if you get a bad gene, hopefully your breaks happen. And so you can prevent and you just have normal growth. But if your breaks fail, then you have loss of the normal growth and you have the abnormal growth and then a tumor. So remember in video 1, right? Remember in video 1 for this chapter, and I was using this as showing you about cell signaling, right? And this is one of your highly suggested reading and thinkings as well, to phosphorylate retinoblastoma, the RB. Okay, this one right here facilitates the cell cycle because it ends up releasing, if you have the CDK2 and cyclin E, it releases E2F, which promotes the S and the rest of this cell cycle. So RB is involved in retinoblastoma, but it's also involved, this sequence right here, in breast cancer, prostate cancer, bladder cancer, and small cell lung carcinoma. All right, and then a major player is a P53 gene, okay? P53 gene, normally, like let's take a look at this scenario right here. UV light, damaged DNA, you start your signal transduction pathway, remember your protein kinases, and normally you would get an active form of P53 and you would repair that DNA, which would inhibit the cell cycle. But if you have a mutation in your P53 gene, even though you got the signal that said, hey, we got a problem, we're out of control on this cell, somebody's stepping on the gas, please quickly put on the brakes. Here, your brakes are broken. Okay, you have a defective or missing transcription factor such as P53, and as a result, you can't step on the brakes. Okay, and in this case, okay, no active P53 means no brakes. So P53 suppresses cancer in 4 different ways that I want to identify. Okay, 4 different ways. So here, first of all, look where it is. It's right here between going from G1 to S. Remember in our regulatory, like this would be like going through airport security, right? So in 4 different places. So 1, it activates P53, activates P21, and that is a gene that binds with CDKs, giving them time for DNA repair. It's saying stop. Okay, it's stopping CDK2 and saying let's not move forward into the S stage. Let's allow time for repair. Number 2, it activates a group of microRNAs, which inhibit the cell cycle. Number 3, it turns on genes directly involved in DNA repair. And number 4, it activates your suicide genes. Remember the caspases I talked to you about? Okay, from video 1, it activates suicide genes, caspases to stimulate apoptosis. And it is estimated that, are you ready for this? Over 1/2 of human cancers are due to abnormal or deleted P53 genes. 1/2 of all cancer. If we could just figure out how to fix P53, half of all cancers, done. Swipe right, or whichever way you're supposed to swipe, okay? Now, the origin of cancer, okay, is a failure of DNA repair system. That would be one origin, failure of DNA repair systems. And then another one is telomeres. Now, telomeres are at the end of chromosomes, and every time you do mitosis, that end of the chromosome gets, and it's not coding for any proteins, okay? But they get shorter at the end of each cell division that you do. But there's something called telomerase, okay? And if it keeps rebuilding the telomeres, remember ACE is an enzyme, right? If it keeps rebuilding the telomeres and they don't get shorter, then it's like an immortal cell. It keeps replicating. So telomeres are at the ends of chromosomes. They're not shortening with each replication due to telomerase, an enzyme that keeps the chromosomes at a constant length. Cells with short chromosomes tend to stop dividing. So these don't stop because their chromosomes keep getting rebuilt. All right. Now, these I'm just going to go through quickly because they're important, I think, for people to understand because cancer is prevalent. There are no notes on these. It's just a quick rundown on these few things. So one is the cause of cancer. How could you get it? Something like breast cancer genes, that's through your genes, okay? So you can't control that. Your DNA is your DNA. However, you can be hyper aware that you have possible cancer-causing genes, so you are more aware that that could be something that you need to look out for. Then it's self-induced cancer. There are some cancers tied to being overweight, obviously smoking, things like radiation, whether you're exposing yourself due to tanning or the jobs that you do, that would be another cause of cancer. All right. So if you look at influences that would cause proto-oncogenes to become oncogenes, right? So it could be heredity, radiation sources, pesticides and herbicides in your environment can cause that. And even they're looking at some viruses as transmitting from host 1 to host 2, an oncogene or a regulatory factor. So what do? You get screenings. So this is a joke, but not really. You can get mammograms, right? Regularly get mammograms when you get a little bit older, right? To check and look for tumors. When you become sexually active, you need to have those, especially things that you cannot see, right? You need to have that looked out for. You need to get regular pap smears in order to double check to make sure that there are no cancerous tissues there. You can get tumor marker tests. This is to see if it's in your DNA or if, oh, sorry, that's the next slide. This would be if it's in your DNA, you can get genetic tests to see if you're inheriting it. But cancer cells will give off certain proteins. And so you can look and have those tested to see, like, if you have prostate cancer, they can check your blood. That's why you want to get regular checkups, because they can run tests to see if any of those proteins are in your blood. It's important if cancer runs in your family, if you have family members who have passed away due to cancer, go get your DNA checked to see if you are potentially a carrier of those cancerous genes as well. Now, let's say you do find out, you can have a biopsy done to check to see if that tumor is spreading or not, or if it's cancerous or just a fatty tissue that is building up. And so there are other preventative measures you can do. Okay, those were the bonus slides. Now back to the notes. I am at 9.5, prokaryotic cell division. So what I taught you before about mitosis, prophase, metaphase, anaphase, telophase, cytokinesis, that is true of eukaryotic cells with linear chromosomes. Prokaryotic cells do not have a linear chromosome. They have a single circular chromosome, and that single circular chromosome will replicate, and then that cell will separate, and that is called binary fission, when prokaryotic cells replicate, and they do this through asexual reproduction. So on your notes, prokaryotic chromosome is a single circular chromosome located in the nucleoid region. Remember, they don't have a nucleus or other membrane enclosed organelles. And it's asexual reproduction. Offspring are genetically identical to the parent. Genetically identical to the parent. This is called binary fission. Chromosome replicates first, then cell elongates and builds a cell membrane and wall between the two identical chromosomes. All right, and then here is just a little chart to compare and contrast prokaryotes and reproduction and eukaryotes. And I filled in your chart already for you there at the end of your notes. And here's just a little summary of what happens during the cell cycle. So don't forget to go back and look at that in video 1. And you know what? That's the end. So hope you guys are having a, I can't stop it, are having a great day. Okay, that's it.